So I'm going to talk about metabolic flux analysis in cancer. So I would like to start my talk with a basic question. What do cells need to proliferate? People in cancer field tend to focus mostly on energy generation, which is the APP production majorly through glycolysis pathway in cancer. But what left behind is the micromolecule synthesis, like protein, lipids, and nucleic acid, which precursors are made in mitochondria by T-cell cycle. And how to cell to replace T-cell cycle intermediates is supported for using biosynthetic pathways. You use the process called anaphoresis, which means filling out the pool. And we've learned that in most of the cancer cells, they prefer to use glutamine for anaphoresis, as the blue line shown here. Our lab is interested in cancer metabolism. We use both metabolomics and flux analysis to address our question. So what's the difference between those? Metabolomics only allowed us to look at the static status of the metabolite, like identify which car is in the parking lot. But flux analysis allowed us to trace the metabolite movement along with the pathway. Like the picture showing here, well, if each car represents a metabolite, one could think that at the left limb, there's more car, therefore the activity is higher. But the truth is there's the stop line causing all the car stop there. And on the other hand, the right lane is actually the car driving faster, which means the metabolite is flowing more actively. Only flux analysis are able to distinguish that. So how do we do that? We use stable isotope labeled nutrients like glucose and trace the metabolite by two complementary methods, mass spectrometry and MMR spectroscopy. Glutaminase is the major enzyme that converts glutamine to glutamate. It has been shown to be upregulated in many kinds of cancer. However, the mechanism remains poorly understood. So we asked a simple question: what will happen if we knock down glutaminase in tumor cells? In our favorite model, glioblastoma cells, upon glutaminase suppression, we found there's no change in glucose consumption or lactate production, but glutamine usage is significantly lower. We also found glutaminase suppression impaired tumor growth in two-dimensional culture or in xenografts. Therefore, we conclude that glutaminase is required for maximum growth of glioblastoma cells in culture and in vivo. However, the residual growth of the glutaminase knockdown tumors made us wonder whether another alternative pathway is being turned on. We then carry out the metabolic flux analysis using C13 labeled glucose. And we found there's more glucose-derived carbon into the T-cell cycle intermediates. But what's more interestingly is we found there's an increase in citrate N+2 labeling. Among all the possibilities, we found the most compelling explanation is that cells turn on pyruvate carboxylase and use the carbon derived from glucose. So if cells can use pyruvate carboxylase, we will be able to see one label on acylic acetate, and that will condense with another label with acetyl-CoA, give you a +2 labeling. Since GC mass spec cannot distinguish which carbon is being labeled, we carried out MMR spectroscopy to address this question. If PC activity is higher, we will expect to see an increased fraction in doublet 2,3 and quartet. And indeed, that's what we found in the glutaminase suppressed cells. Those peaks do goes up. And those peaks goes back down when we co-deplete the cells with PC. We also validate this with another independent assay. Together, the data suggests that glutaminase silencing increased glucose-dependent anaphoresis through PC. We then asked whether PC activity will be upregulating in a glutamine-addicted cell line when we adapt them to grow in chronic glutamine-deprived condition. As you see here, adapted cells now can grow with no glutamine, while parental cells are pretty much dead. We're then developing an assay to directly measure PC activity by using C13 label pyruvate on one position. As the diagram shows here, only through PC reaction will give rise to a N+1 on citrate. And as the results show here, the adapted several cells have higher PC activity upon glutamine withdrawal. We then ask if these cells now can survive without PC. And the results show here now they can grow really well. Therefore, we conclude that PC activity is induced and required for cell growth during chronic glutamine deprivation. We also found that in some cancer cell lines with higher PC basal level, they can grow without glutamine, and targeting glutaminase in those cells has no effect on their cell growth. Therefore, for people who are thinking about targeting glutamine metabolism as a potential cancer therapy, they need to take our finding into consideration. So to briefly summarize this part of my talk, I hope I've convinced you that when glutamine metabolism is impaired, cancer cells, however, can figure out another way to compensate by rerouting the carbon from glucose through PC to replenish TCA cycle intermediates and support their growth. Now I would like to switch gears to talk about a current project that I'm working on, which is imaging metabolism using dynamic nuclear polarization, or so-called hyperpolarization. MMR using stable isolab has been routinely used in whole body studies of human metabolism. However, the major drawback of this assay is the low abundance and sensitivity of the nuclear spin, resulting in a low signal-to-noise ratio. Hyperpolarization has improved the sensitivity by 10,000 or more, and that drastically increased the feasibility of real-time MMR-based imaging of metabolism in vitro or in vivo. So how does this technique work? In principles, pure electrons were doped with the C13 material and placed in near absolute zero temperature and high magnetic field. They will be highly polarized. And then the polarization can be transferred from the electron to C13 nucleus, resulting in high spin status via microwave irradiation. So by using the hyperpolarized pyruvate, we are able to look at various downstream metabolites within seconds. So we use hyperpolarization technique to analyze tumor cells, but I'm going to show you today is using this technique to imaging metabolism in perfused mouse liver. We use a VHL knockout mouse model, which is generated by Dr. Bugnola's lab. And VHL is a tumor suppressor, which encoded protease, the subunit of an E3 ligase complex, targeting HIF1 alpha for degradation. Surprisingly, they found the liver-specific knockout resulting in a severe mitochondrial oxidation defect. So the way we do that is we inject the mouse with adenovirus Cree into a wild type or a flax mouse. 4 days later, isolate the liver and perfuse them with axinoate, which is the fatty acid. Following by injection of hyperpolarized pyruvate, then we can acquire data in real time, real fast. So here, as you see here, 20 seconds after injection of hyperpolarized pyruvate, we're able to see the maximum production of lactate in the wild type mice. But this peak is much lower in the knockout. And if you look at a sum up of 14 scans, and the spectrum will show more metabolite, but the profile of two livers is very different. So we're still working on the mechanism for that, but with no doubt hyperpolarization provide a faster and much more sensitive way to measure metabolic fluxes. To summarize my talk today, in the first part, I've shown you that profiling metabolic fluxes in glutamate-suppressed or chronic glutamine-deprived cells reveal induction of the compensatory and a polarity mechanism catalyzed by pyruvate carboxylase. This finding may prove to be a mechanism of resistance to therapeutics targeting glutaminolysis. The second part, we proved that hyperpolarization enables a real-time MMR-based imaging of metabolism in cells, perfused organ, and ultimately whole animal. And that could potentially apply to clinical diagnosis of metabolic diseases and cancers. Lastly, I would like to thank all members in Deborah Dennis lab, people in NRC, Buglores, and Matisse lab, and all the funding grants support our work. And thank you for your attention. I'd like to take any questions.